cell is ready to start dividing, and then from here you have what we call these little spindle fibers that gather here, and then each cell now, each little chromosome develops a little centromere, and it begins to divide. All right? So now we have this little chromosome, all right, that divides by itself, and it goes into this, and what we call a little sister chromosome. And it has what we call a centromere. All right? And this has what we call, anybody know what this is your DNA, your double helical structure, because you've got one for this and one for this, all right? So out of one chromosome, we make two chromosomes. And they're held together by this little centromere. And then this little sister chromosome divides into, okay, what we call daughter cells. Or daughter chromosomes, all right? All right, and then what happens is this little chromosome now, once it does that, it's ready to go into the different stages of mitosis, which is like prophase 1, prophase 2. So in prophase 1, this is what's happening. In prophase 1, we're losing the nuclear membrane of the cell, all right? And this dividing is going on, and the chromatin is gathering up to chromosomes, all right? So we have different steps that are happening. In prophase 2, hey, those little chromosomes, they begin to self-divide, and now we have from one chromosome a sister chromosome, all right? We have now, let's say, metaphase, and in metaphase, okay, here we are in the nucleus, all right? We may have the little chromosomes now sort of gathering itself up into what we call the equatorial plane. So we have so many of them that are going to be gathered by the spindle fibers here, all right, so that the cell can now begin to divide, right? And then, let's say over here, we may be in anaphase, and in anaphase, all right, that cell now, we got chromosomes going over here, and we have two chromosomes going over here. It's still not dividing, but sort of working itself. And in telophase, okay, you have one here and one here, with 46 chromosomes here and 46 over here. All right? But what happened? We have another thing that occurred. Inside the cell, we have what we call cytoplasm. The cytoplasm is a gelatinous protein substance that acts like a buffering agent so that all the little organelles inside, like the nucleus, the mitochondria, the endoplasmic reticulum, all those little guys don't bump into each other. So the nucleus goes through a different process. It sort of furrows, sort of begins to furrow in like this, like taffy. You know how taffy goes like this? All right? It twists like this and goes out like that. All right? So the cytoplasm divides differently, all right? It goes through another division. And right now the word escapes me as to what it is. Does anybody know? Nobody can tell me? What? Oh, cytokinesis, that's right, cytokinesis. I couldn't think about it. So cytoplasm divides into what's called cytokinesis. All right? And that is the furrowing of itself before it splits. So you have a cell going into the cell cycle, differentiating, becoming a cell of whatever it is. It could be a cell of a hair, or a needle of a porcupine. It could be whatever it is, all right, or an elephant's tusk. Because all eukaryotic cells go through this process, all right? All mammals go through this process. So we have all these divisions. They go in there and they come out. They do this division They go into cytophases, and there you have it. All right? That is mitosis. And mitosis is going from 46 chromosomes to another 46 chromosomes, making an identical cell of itself. Okay? In meiosis, all right, what we're doing though is we have two cell divisions, really. We have meiosis where we have the chromosomes exchanging as they are doing this, you know, sort of like dividing, finding themselves so that they can make a sister chromosome like this. What they do is we're trying to get the cells to go through their first division, in which this happens, but at the same time we have some particular things that happen. In meiosis, we have something called crossing over. And before the cells ever get to this state, all right, they're in their growth state where there are little chromosomes inside the nucleus. Each chromosome sort of like binds to itself, all right, and then what happens is they exchange information, and when you have the chromosome, you have two chromosomes. This chromosome may take this little bit from, it may be like this, all right? In other words, let's say, let's do it better. Let's say this is this chromosome and this. Well, let's say now, when they crossed over, this chromosome may took a little bit of this part of this chromosome, and then this chromosome gave a little bit to this. All right? So we call that meiosis 1, and that's the first cell division. Then, once that happens, it goes through regular mitosis. And once we go through regular mitosis, we do the second mitosis. We need to do another one in order to go from 46 chromosomes to what? 23, which is a haploid number. You go from a diploid to a haploid. I think a lot of you know this, correct? Many of you should know all this part. This is just biology, but this is what we're going to sort of like do a little overview. Any questions on this? Do you feel confident you know what's going on here? I would think so. It's all about life. Okay, so given that the only other thing that I would say is important is that to give a contrast, they talk about eukaryotic cells here, animal cells. Well, prokaryotic cells are what? They're your bacteria, all right? They're your viruses and all that. They go through acellular reproduction. From one cell, another cell emerges. They don't need to have two cells. They don't need to have a sperm and egg, all right? They just need to be who they are, and they'll divide through fission, cell fission, all right? So they'll divide, and they'll make two of them. That's how bacteria works. You got one little strep, you got millions of strep within 24 hours, all right? It's how that works. Okay, are there any questions on this? Any thoughts? Because this is the basis of this chapter. If there aren't any, then I would say we can go to lab and start doing the leaf lab. The leaf lab has to, it's a long lab, or the first one is long, but I think we'll do the first two. First experiment and the second one.